Hello, everyone. Welcome to this webinar on nematodes in practice from research to field application. Thank you very much for taking the time to join us today. This webinar is hosted and presented by CABI, the US Department of Agriculture, Natural Insect Control, and BASF. It is being recorded. Please take a moment to introduce yourself in the chat. Tell us who you are and where you're from. We're very keen to know who's joining us today. And we encourage you to ask questions throughout the presentations using the Q&A function. We'll also be keeping an eye on the chat, but please use the Q&A function for questions. We will have time for questions and address them at the end of the presentations. And at that point, you can also use the raise hand function and ask a question verbally, but I'll remind you when we get there. So thank you again for joining us. We hope that you enjoy this webinar. And with that, I'll hand over to my colleague, Dr. Stefan Toepfer from CABI, to start us off.
Okay, welcome everybody to the webinar on nematodes in practice. We see lots of participants, probably from all over the world. So a good morning to the Americas, a late afternoon, good afternoon to Europe and Africa, a good evening to Asia, and a very late night, good evening to the Pacific. So we are happy to see you online here in big numbers. As it was already said, this is the webinar on nematodes in practice from research to field application organized by the Bioprotection Portal, and we will have speakers from BASF, USDA, and NIC. So the aim of the webinar is to strengthen knowledge and trust and capacity for the use of entomopathogenic nematodes as a biocontrol tool within the North American IPM systems. I have one slide here on what are nematodes. Many of you are very much familiar with this, but let's have a quick look here that everybody has the same understanding. So they are tiny worms, roughly 0.5 millimeter. You can see them with a hand lens actually, but otherwise they are rather tiny. They're usually soil living, so they are soil organisms in most cases. And this group of nematodes parasites insects, but they have some symbiotic bacteria which are pathogens to insects. So that's why we call them scientifically entomopathogenic nematodes, EPN, which is quite a complicated word. Sometimes we call them entomoparasitic nematodes because they behave like parasites. But simply, we very often use the term beneficial nematodes when it comes to biocontrol in practice. Just to note that these EPNs, these entomopathogenic nematodes, have nothing to do with plant parasitic nematodes. Many of you are probably very much familiar with the pests, the nematode pests to crops. There are also human nematodes. There are lots of saprophytic nematodes which actually help to decompose organic material in the soil. But they're all very, very, very different to entomopathogenic nematodes. Nematodes themselves is a huge group of organisms, and they have different orders, different families, and these entomopathogens are very specific and have nothing to do with the other nematodes. So they are used in biological control because they kill insects, right? They are the third most used group of biocontrol agents globally after Bacillus thuringiensis. Many of you know BT as a biopesticide or transgenic and so on. And trichogramma parasitic wasps are very frequently used all over the world. But then already the nematodes are coming in. So they're quite important biocontrol agents. But there are some misconceptions and misunderstandings, and I will list here a few without giving the answer because we will try to give you the answer during this webinar. So some people may state EPN may attack my crop plants. We have already answered this actually, they will not. EPN are rarely effective under real farming conditions. EPN products are not available. EPN can be sprayed like an insecticide. EPN cannot be used together with a synthetic pesticide or in alternation with pesticides. EPN are only for organic farming, not for IPM and not for conventional farming. And there are many other misconceptions, and there's also some misunderstandings. Certain things are not clear. So we will try to solve some of these issues here today in the webinar. So we're having 4 speakers and four talks. The first one is scientific foundations and implication for use, which will be provided by myself. I'm scientist in CABI. I'm also crop health advisor and have been working for many years on entomopathogenic nematodes. Then we have a talk about public research, public sector research and field evidence by Dr. David Shapiro-Ilan from USDA, Georgia. Then we have practical application from theory to field by Stacey Hickman from Natural Insect Control, Ontario from Canada. And then we have integration of beneficial nematodes into IPM and their use in resistant management by Diana Londoño from BASF. And we have a question and answer session and a discussion, which is hosted by Pia Eliason. She is the project manager for the CABI Bioprotection Portal from CABI. So again, I would like to welcome everybody, and I would like to immediately start with the first presentation. So the first presentation is about scientific foundations and implications for use. As I said, that's presented now by myself as a crop health advisor from CABI. CABI is an international organization, not for profit. It's intergovernmental and is trying to solve issues in agriculture, food security, but also food safety. And when it comes to food safety, of course, the entomopathogenic nematodes play a big role. So let's start with the nematodes. I will go step by step through the life cycle and try to make the link to the usage. So what does it mean for using nematodes? So first of all, nematodes are usually soil living. They are in the soil. They are living there naturally, sometimes not in high enough numbers, especially not in the cropping systems, so that we would like to add them to the soil. So we use them, we buy a biocontrol product and add them to the soil. But they are naturally already there. And in many systems and many habitats, they are very effective, but in these disturbed cropping habitats, sometimes they are not enough. So we add more. Then these nematodes can search for the insect and can enter into the insect. There are very few, 2, 3, 5 nematodes only needed to enter the insect, but still nematodes need to be applied in big numbers. Imagine you have the soil, this is a 3D system, it's a volume, so you need quite a lot of nematodes in the soil so that they can find and attack and enter an insect, although very few are actually at the end enough to enter an insect and kill it. Then there's something special. They have bacteria, they release the bacteria, and the bacteria are toxic to the insect, and the insect is dying. So now this process takes 2 to 4 days. Yeah, entering of the nematode, the bacteria start to multiply and kill the insect. So it's not a knockdown effect. It takes 2 to 4 days. Of course, depending on the cropping system and the nematodes. And if we apply them artificially through spraying or injections and so on, of course, they need also a bit of time to find the insect. So it can take up to a week until you see the effects. There's nothing bad about this, it's just important to know that there's no knockdown effect by a nematode or by its bacteria. Then the nematodes multiply. Yeah, they grow and multiply, although only 2 to 3, maybe 5 nematodes have entered, you will have thousands at the end in the insect. The insect is dead, and once the insect is eaten up, the nematodes exit into the soil. And you see on the right side a picture where you have this, if you look carefully, you see tens of thousands of nematodes coming out. They will live in the soil and can infect the next insect. This is, of course, an advantage of biocontrol. Biocontrol agents often can multiply, what synthetic pesticides cannot do this. So there are quite some implications here for the usage when we try to understand how these nematodes work. I have sorry, one more slide, one more line, used in biocontrol. So we are using in biocontrol this stage which is in the soil. This is a young stage of the nematodes. It's quite strong. It does not feed. It's well protected, except against drought and UV light, but otherwise quite well protected. And this is what is sold in a product, in a biocontrol product. So what you buy and then apply. So I have two more slides for implications of use. So nematodes function implication for use. Nematodes can actively search for an insect, so like a parasite to it or like a predator is doing, but a synthetic pesticide cannot do. Many biopesticides cannot do, but nematodes can actively search for the target. They are often applied like a pesticide or biopesticide, but there's no contact spray needed. So the nematodes do not need to be sprayed onto the insect, like a contact pesticide, for example. There's no ingestion needed. So sometimes you may, if you remember, there are some biocontrol agents, biopesticides like viruses, bacteria, they often need to be ingested. So the larvae must feed, otherwise it will not work. For nematodes, no, they can search themselves, they can enter themselves, the insect. However, there's lots of water needed. So it's not really true applied like a pesticide, partly like a pesticide, but with lots of water. And nematodes can be alternated with many pesticides. I said already the free living stage of the nematodes is not feeding. It even does not have a mouth. So nothing can enter really the nematode. So they are quite strong when it comes to protection from chemicals. Not to every chemical, but quite a lot. We will have today a talk about this. It's one of the topics of the talks today, the compatibility with other IPM tools like synthetic pesticides. Good. They are living organisms and they can only work if they stay alive. So this is, of course, a challenge sometimes, especially nematodes need to be cooled during storage and also during transport. If this is not happening, the nematodes may die and may fail when you apply them. Moisture is needed. Nematodes can only survive if it is moist. If they are sprayed like a herbicide and are drying on a leaf or on the ground, they are dead. Then I said this already, a few nematodes actually only needed to enter an insect or to kill an insect, but still they are applied at huge numbers, the nematodes, because you have to fill the soil or the target area, the soil, the bark, and so on to get consistent results. I have down there two lines of dosages, which are very rough estimates, like 2.5 to 5 billion per hectare or 1 to 2 billion per acre. You will hear more about this in the other presentations today. It depends, of course, on the crop, on the target pest, and so on. So these numbers you see there are very rough estimates only. Good. How to find the right nematode product? It depends on the target pest, the target stage, the crop, what application technology you have available on a farm, and which products are available in your country. So target pest and stage, yeah, nematodes are used against insects, not against mites, so not against spider mites and so on. So they are used against insects. Nematodes are usually not highly host specific. They are group specific. So there are nematodes which will kill beetle larvae. There are nematodes which will kill fly larvae. So they have a wider usage range than some other biocontrol agents, but they have a narrower range than a broad spectrum pesticide. So you cannot use one nematode against everything. So there are different species of nematodes, and you can kill certain groups of insect pests. They're usually most effective against larvae, but there are some examples where you can use them against adults or pupae, for example, pupae of thrips and so on, but usually they are very effective against larvae stages. And they're usually used against hidden pests because pesticides cannot really reach pests which are hidden in the soil, in the roots, in the tubers, and nematodes can find them. So here's a big table. I don't want to go too much into detail, but on the left side, you will find some nematodes. They are very complicated names like Heterorhabditis bacteriophora, Megidis, and so on, Steinernema capocapse. And then you see target groups. So it's not necessary that it's not the case that Bacteriophora works only against one species. No, they are very good against grubs. Megidis is very good against weevil larvae. Capocapse is very good against Lepidopteran caterpillars like cutworm and so on. So you see here that the usage range is quite big. And I have another table here, Steinernema felsie, glasery, and so on. These are another few species of nematodes, and they are also group specific. So you can use the Steinernema felsie against maggots, all sorts of fly larvae. The same for the other ones. They are group specific, which helps a farmer quite a lot because felsie can be used against several types of Diptera larvae, for example. Good. How to find the product? Yeah, we have the CABI Bioprotection Portal. It's the world's largest free online source for bioprotection products. Um, you can go online, you can search for products. They are at the moment for 54 countries online. When it comes to North America, yeah, Mexico, US, and Canada are online. About 7,000 products and biocontrol products, sorry, bioprotection products, and around 145, 150 products when it comes to nematodes. But only 6 species are commercialized, but they have many, many products. So, for example, the first 3 you see there, Steinanema felsie, Heterorhabditis bacterfa, Steinanema capocapsa, there are many products. So these are the classic nematodes which are available in many, many countries from Asia to the Americas, Europe, Africa. You have these products available. And then there are some more specific products. So you find them on the portal. So you can search. You can search for the country, for example, Canada. You can search a crop if you want. And I was searching here now for fungus gnat larvae. So these are Diptera larvae. They attack, for example, mushroom production. And then you get products. I'm not talking now about the product names and so on, but you will see that there are some nematodes, Felsie, Capocapse, and so on. And you see these products, you can click on them and you get more information. You get the product label or you get information how to apply them. So for these fungus gnat larvae, this is for mushroom production because you cannot really spray mushrooms with a chemical because you will eat them. Also very common in greenhouse ornamentals and vegetable transplants, seedlings, and so on. And we will have a talk today which will explain the use of these nematodes against these small maggots, actually. And most of the products are based on Steinanema felsie. So you cannot really use Steinanema causii against these kind of larvae. You need a lot of water. You need a moist medium. In greenhouses, this is relatively easy. And then you apply the nematodes in water. We will hear more about how to apply them in a talk later today. I will give you only one more, 2 more examples. There are the rootworms. You know them very well from North America. You can search for rootworm. You will find a nematode. So Bacteriophora, Heterorhabditis bacterfa is the key here. And many of you know cutworms, all sorts of crops. You can also search here and you will find Steinanema capocapsae. So there are nematodes available, especially for these kind of soil pests. Application technique may be a bit difficult for rootworms and corn. Corn is a field crop, it's a large scale crop, so you need special equipment like soil injectors, the sowing machine. So when you sow the maize, you can apply the nematodes as a liquid in the moment of sowing. For cutworms, it depends what cropping system, whether you have problems in berries and vegetables and turf grass. So quite often applied with irrigation, but it depends now really on the crop. You need to get the nematodes with moisture into the soil. Good, these are 2 examples. We will hear more examples today, how to use nematodes in practice. Please remember the bioprotection portal can help you. It can help you to find products. It can help you to find information also how to use the nematodes. There is a resource section on the bioprotection portal where you find lots of information about biocontrol. And of course, for each product, there's product information. So we hope that it helps advisors and farmers to make informed decisions for still sustainable pest and crop management. And we hope to improve also the credibility of biocontrol through data, through success stories, and to have through a reliable source, which is here the bioprotection portal. So I'd like to thank you all for the attention, and I will hand over to my next speaker, which is Dr. Shapiro-Ilan from USDA RS, talking about public sector research and about evidence that the nematodes work or under which conditions they will work. Thank you.
Sorry about that. I was looking for the unmute button. Okay. Is the slides, everything's look okay, Stefan?
Yes, looks good.
Okay. All right, thank you. Yeah, I really appreciate the opportunity to speak here today. I'm really going to be talking about just a few approaches to improve the efficacy of entomopathogenic nematodes as biocontrol agents in the field, and then a bit of learning from trial and error that I've experienced, and I'm sure many of you have had your own experiences in that. So I'll just going to start out with a bit of the drivers. Why do people use entomopathogenic nematodes? And then what are some barriers or limitations to using these entomopathogenic nematodes, or we call them, as we call them, EPNs as well. A bit of the lessons learned from my lab and field trials. Then I want to focus a bit on several examples of our recent advances to improve formulation application technology, and then just a bit of broader horizons for biocontrol that I'll be getting into. So what are the drivers? Why do we use these nematodes? What are the beneficial attributes of these nematodes that make them so attractive as biocontrol agents? The nematodes, as mentioned, are really safe to humans and other non-targets. They're compatible with organic practices, or at least I think most products are. Some formulations may have some incompatibilities. They generally kill a wide range of hosts relatively quickly, especially compared to some other entomopathogens. There are a couple of specific nematodes, Steininium scarabi focusing on white grubs and Steininium skepterisci focusing on mole crickets, but they're not so much commercially available at this time. This durable infective stage, the infective juvenile IJ, really makes them amenable to mass production and application using standard equipment. So the farmers can apply the nematodes generally the same way they apply other tactics such as chemical insecticides, most agricultural spray equipment, and most irrigation systems. So their extreme safety is well recognized, so much so that the nematodes often have a special status in terms of regulatory pressures, so that they're, for example, in the US, exempt from EPA pesticide registration. So with all of this and the fact that, as Stefan mentioned, they're used to control a wide variety of economically important pests, we see EPNs as a success story in biocontrol. But we also see that there are barriers to expanded use of the nematodes, such as cost, especially in comparison to some of the broad spectrum chemicals. The nematodes are sensitive to the environment, to heat, desiccation, and relative humidity, UV radiation. The nematodes have sometimes been cited to have inconsistent field performance. And the storage requirements, as mentioned by Stefan, can also be a barrier in terms of having to keep the nematodes in cold storage makes it difficult for distributors to keep the nematodes and for farmers sometimes to keep and apply them. Then we have some basic gaps in knowledge of the nematodes, which we need to fill those gaps in order to better understand the nematodes and use them better in biocontrol. So we think about what are the approaches to developing and improving microbial control programs with these nematodes. The first step, building off of what Stefan said, is identifying the candidate organism. What's the best nematode to use, the best species, the best strain? And you may get high levels of efficacy, sufficient levels of efficacy, just with that choice of candidate organism. But if not, there are approaches to get better efficacy, and one of those is to improve the organism itself, improve the nematode, or improve the production, application, or formulation technology associated with that nematode. You then would go into kind of analysis and feedback. How can we make this even better? And never forgetting your basic research that we could then leverage to get better biocontrol. And always we need to look at the use of these nematodes in the broader context of IPM programs. Sometimes the nematodes will be excellent standalone products. Sometimes they're going to be integrated with other products either simultaneously or on time. So let's talk a bit about identifying the candidate organism. This is usually done by initial laboratory and greenhouse screening of a variety of strains and species, and then followed by field testing of the most promising ones found in the lab. We'll also need to use our literature and knowledge of the pathogens and target pest. So for example, you may already know that you're not going to use nematode that's adapted to high levels of heat in a cold situation in the field. And this, again, identifying the candidate may lead to success all by itself, but sometimes you need to go further than that. And let's look at the example of plum cuculio. Plum cuculio is a major pest in the U.S. of stone fruits and pome fruits, so apples, pears, peaches, plums. The adult insect attacks the crop directly, larvae drop to the soil, and then the adults overwinter in around the orchard. Early pathogenicity of nematodes was found in the larvae, so that we decided to screen a wide variety of nematodes, most of these commercially available, for which one's going to be the best. And this was a laboratory study. And we see that SF, or S. feltii, was the very best one, and followed by Esria brava. So these are the two that we tested in the field. And in the field, we found that Esria brava provided outstanding control of plum cuculio in the soil, whereas S. feltii was not effective. And we think that this was likely due because of the heat conditions in Georgia in peach orchards, which is where we tested it. So this is a nice example, really, where the lab results did not translate to the field. We expected first S. feltii to do great, but it didn't. Nonetheless, we have Esria brava as a great control mechanism for plum cuculio, and also S. carpocapsia is since found to be highly efficacious. Another example would be against peach tree borer, which is a Ceciad pest, clearwing moth. The larvae penetrate deep into the roots. We can see great control with S. carpocapsia. We screened a bunch of nematodes to find that one, and in this case, we actually got great control right away, so there's no need for further investigation. But what if you don't get great control to begin with? You can improve that biocontrol agent, that EPN, through these methods. You can improve the strain itself, and you also need to stabilize that strain, so once you get a great strain that it doesn't deteriorate. You can improve the strains through genetic selection, hybridization, creating purebred inbred lines. You can then improve the mass production to get better quality nematodes, lower costs, improve formulation and application technology. We can also look at environmental manipulation, which doesn't actually change the application itself, other than you're manipulating the target area, the environment, to make it more conducive to the nematodes so that they can get greater efficacy, such as, for example, maybe having a mulch or some kind of ground cover that protects the nematodes from UV radiation. That would be environmental manipulation. And again, let's not forget about understanding biology and ecology so that we could leverage new knowledge to improve efficacy. So I'm just going to give a couple of examples of improving formulation and application technology. So let's take the case study of lesser peach tree borer. Probably the biggest barrier to wider use of entomopathogenic nematodes is in applying them to above ground pests. And that's because of their sensitivity to UV radiation and desiccation. So if the lesser peach tree borer attacks peach trees and other stone fruit trees on the scaffold limbs and trunk, causing wounds like shown here. If we apply nematodes just in aqueous suspension, they're going to fail because they're dying in that heat and desiccation and UV radiation. So we have to protect the nematodes. And well, we screened a bunch of different formulations and came up with this barricade gel. And this graph showing here, SC or S. carpocapsia with barricade at 2%, which means it can go right through the sprayer or the full rate where you apply the nematodes and the barricade on top, and you'll see a thick gel like shown here on the peach limbs. These did great in reducing the percent live lesser peach dew borer, but nematodes alone you can see failed, not different from the control. And our nematode treatments did just as well as chlorpyrifos, which was the standard chemical at the time. So this gel can really help in application of the nematodes. And you could also use a gel like this in soil, especially if there's no irrigation, to protect the nematodes from desiccation. And we saw this with peach dew borer, which is different from lesser peach dew borer in that it goes down into the scaffold roots of the peach tree. So it's a soil pest, basically, not above ground. We get very high levels of control with S-carpocapsae, very economical because you only have to apply around the base of the tree because the insect goes straight down into the roots. The only problem is if you lack irrigation, as many peach growers in the US do, barricade can be a substitute and you apply it around the tree. It doesn't have to be hand applied, it could be tractor applied. So you can see here the nematodes with barricade did just as well as chlorpyrifos, but the nematodes with no irrigation fail. So again, you can use that gel to the soil too. Sorry about that. Barricade is a great tool, but it's a bit expensive and can be difficult to use. So we've been screening other gels, other formulations to protect. This work being done by Sabrina Elgar and Brett Blau's lab at University of Georgia. We see our best ones. We were screening these in outdoor tests, which ones protect the nematodes the best. The barricade, carboxymethylcellulose, which was developed first by Ted Turling's lab, xanthan gum, and sodium alginate, all did great. And so these are ones we want to look at further for their lower costs as well as ease of use. Another lesson we kind of used when we took those to the field against peach dew borer, we wanted to see which one of those formulations I just mentioned is the best one. But 2 years in a row, we got really a lot of rain. So this just emphasized, as you can see, even the nematodes with just water, which would usually fail, as I showed you before, did great because the soil was moist and all the formulations didn't differ. And that they all did way better than the control. So this just emphasizes you don't really need a formulation in that peach system if you have wet soil. Another formulation that we're working on, and it's in the earlier stage, is nanoparticle formulations. I think of this as really cool because it's kind of like a coat of armor on each little nematode. Each little nematode gets these little nanoparticles around it, which protects it from UV radiation and desiccation. So we're testing this, showing that they protect in the lab and initial field tests. And recently we found that various nanoparticle formulations are effective, titanium as well as some natural protein-based nanoparticles. This work by Abigail Croft, postdoc in my lab. Moving on now to improved application. So one approach we can have to get better efficacy is through what we might call boosters. Something added so that the nematodes will kind of get turned on and infect and disperse better in the field. So here I'm talking about pheromones, ascaricide pheromones that drive nematode behavior. This work being done mainly with Fatima Kaplan from Ferronym as well as Ed Lewis and at University of Idaho. So we see that nematodes talk to each other with these pheromones. And those pheromones drive their movement and infection behavior. And we think also aggregation behavior as well, because we discovered that, very interestingly, the nematodes don't just move randomly in the soil when they're looking for a pest to kill. They kind of move in packs, like a pack of wolves looking to take down their caribou. The nematodes are looking to take down their white grub or whatever. And they're moving in packs, maybe with an alpha nematode in the lead. And we found that we can use their pheromones that direct their behavior to our advantage. We can tell the nematodes what to do and tell them if we expose them to these pheromones, Now it's time to go out and find a host and kill it. So if we expose the nematodes to pheromone prior to release, we get better efficacy, higher kill of the pest. This was shown first with pecan weevil, a major pest of pecan. When we applied the nematodes in December, when it's too cold for nematodes, even S. feltii, which is a cold adapted nematode, you could see the nematode by itself didn't separate from the control in terms of reducing the pest population. But if we add the pheromone, they reduce the pest population. So this was shown a couple of years in a row, and this work by Germain Perrier was a postdoc in my lab now at Mississippi State University. And we also saw enhanced whitefly control with this pheromone approach. So another application method that we've looked at and can give high levels of efficacy is what we call the cadaver approach. Here, and this is based on in vivo production, if you're producing the nematodes and get infected hosts, then you take those infected hosts, those dead insects, and apply them to the target site where the pest is. Then pest suppression is achieved by the nematodes that come out of that insect. And we see that these nematodes have higher efficacy when they come out of the host compared to aqueous application. Now, those of you who've worked with nematodes know that if you apply dead insects, the infected insects to the field, they might just turn into a sticky mess sticking together. So we developed these clay coatings that you could see that protect them from rupturing, sticking together. Another approach we're looking at for improving application is improved persistence, the quest for carryover to the next season. When I talk to farmers about the nematodes, the first question is often, can I just, do I have to apply them again, or will they be able to kill the pests the next year? Because that'll save them money, so they're very interested. So generally, for decades now, most people will say, no, you have to apply them again the next year. You'll find them in the field, but not at the level you need to kill the pest. More recently, people are finding that they may persist, such as with pecan weevil, which 2 years after application, we found high levels of persistence, even with very low rates. So the question is, what controls persistence? How can we ensure that there will be carryover to the next year at the level the farmer needs? Finally, we suggest looking at improved strains, such as developing breeding programs for nematodes, which already has started, and people even developing marker-assisted breeding, where you identify which genes control the important traits, like fecundity, like host finding, like virulence. So we can breed nematodes in a controlled fashion and get better nematodes. So lastly, I just want to touch very quickly on the next step in my career. I'm going to be director of a broader biocontrol lab, not just nematodes, the USDA biocontrol in Montpellier, France. And I'll still have my lab running in Georgia, focused on EPNs and fungi, entomopathogenic fungi, but I'll be taking on for a couple of years this role in France and hope to be able to collaborate with many of you when I'm in this role. And I'll be planning on bringing the nematode technology to France with me. This is a very important lab that focuses on stopping invasive insects and weeds from getting into the US and using biocontrol to stop those pests. So I want to acknowledge all those that provide technical assistance, co-investigators funding, and the organizers of this webinar. This is just a couple of books we came out with recently. The Nematodes as Model Organisms, a broad book from my former postdoc advisor, Emma Glazer and Paul Sternberg, and our book on the EPNs recently from me and Ed Lewis, and many great chapter authors, including Stefan. So that's all I have, and thank you very much. I'll turn it over next to Stacey.
Thank you all. I'm just going to share my screen. Hi there. Hopefully everyone can see my slides. I will start my
I can see
presentation.
them.
Thank you so much. I want to just thank you so much for the opportunity to speak to you today. I'm really excited about it. Thank you, Dr. Shapiro. He has been a mentor to the company that I work for, so we're really great to be able to see him on this call. I'm going to talk about a few things about keeping your nematodes happy, healthy before you do your application and during your application. It's not quite as exciting as dealing with pests, but it is a really important aspect to your nematode journey, using them and being them effective. So I will talk about the handling, the storage, and the application. My name is Stacey Hickman. I work for the company called Natural Insect Control. We are a small family-run business in Ontario, Canada. And we actually rear entomopathic nematodes through in vivo processes, which is a live host application. One funny little thing, I am very Canadian. I probably will say A in this presentation. I also will probably say sorry. So sorry, let's get started. Look at that. I didn't even make it through one slide before I did that. So let's get started. How many of you guys have had an opportunity to use beneficial nematodes? You could just tuck into the chat what number that you are. If you're a 1, yes, I use these on a regular basis. I'm an expert at this. Or I'm a 2. I've used these once or twice, but maybe I need some help in figuring out how to use them a little bit better. Or number 3, no, I'm interested in this. I really want to learn how to do them. Or 4, hey, this is my first time that I have ever heard about nematodes and I want to learn as much as I can. If our amazing CABI organizers, it's an absolutely amazing organization to work with, could give me an idea what kind of numbers they are seeing in the chat, that would be fantastic.
Yes,
My
sure.
idea
We're
behind
seeing, oh, sorry, Stacey, we're seeing lots of answers already come through. So quite a lot of 3s and 2s, I would say. So a good mix though, also seeing 1s and 4s, but probably more 2s and 3s.
I absolutely love that. I love an audience that is nematode curious. That just warms my heart. So that makes me really happy that we've got some good interest, give you some good information on how to apply them and use them. So beneficial nematodes are a living organism. They are not a spray and forget kind of product. It is something that does require some understanding of how to use the product, how to handle it properly, how to apply it properly. It is really important to understand that when you're using entomopathic nematodes. Nematodes have been commercially available since the 1980s, so this is a relatively newer technology, but not really. It has been developed for the last 100 years. So it's got a lot of research behind the products that we have today on the market. So beneficial nematodes are a living organism. They are thought of by most people the same as a chemical because you're doing some very similar activities when you're applying them, but they absolutely are not a chemical. This is a living organism that you're applying to your fields, to your crops. Success really depends on a lot of details that you have to get right. Proper handling, proper storage. We've got to make sure we're properly holding these things in the best conditions possible. Having correct timing for the pests that you're dealing with, even with the environmental conditions that you're dealing with. Using adequate soil moisture. These guys need a lot of water to be able to go down with to find their pests in the soil. A suitable soil temperature. They have a fairly wide range of soil temperatures, but you still need to make sure that you are understanding what that soil temperature is when you're doing an application. And then making sure you do an effective application, taking a look at the equipment that you're using to make sure that you haven't done anything with that equipment that can really damage these nematodes as you're applying them. The most disappointing things that growers see is that the nematode doesn't work and they just think it's that the nematode itself that's not working. And in most cases, it's actually the handling, storage, and application of it. So beneficial nematodes, like I have said a few times, are living organisms. They are not an inert chemistry. They are not a spray and forget kind of product. These guys need to be stored properly, mixed well, and applied well using some very strict considerations with your application equipment. The same product can show an incredible success and also can show a big failure, and that's usually based on the application or the execution of that product. Grower challenges that I see on a regular basis and I hear from my growers is that they are inconsistent in their performance from crop to crop, from season to season. You have to remember that because this is a living organism, they're affected by the environment on the outside. So what is the temperature of the soil? How much humidity do we have? How much UV light do we have? All of those can cause an inconsistency in performance. There's poor timing based on the pest that you have. Maybe it's a pest in a stage that isn't affected by nematodes or isn't present in the soil itself. There's also the weather. Again, nematodes are very affected by UV light and high heat, so we want to make sure that our weather is good. There is storage mistakes. Exposing them to high heat or freezing temperatures during storage is devastating to nematodes. And then there can be application errors. High pressure, fine filters, poor agitation in your tank, all of these can really affect how well a nematode is going to work. And they are a challenge for our growers to be able to make sure that they're keeping all of these in check when they're doing these types of applications. This is one thing that I like to keep in mind when I'm talking to my clients about nematodes is we want to store it right, we want to apply it right, we want to keep our conditions right. So once you do everything right, you're going to find that you're going to have a lot more success with using beneficial nematodes in your crop. So it goes right from the very beginning when you actually receive your nematodes. Coolness is the major factor to keeping nematodes fresh, keeping them viable, keeping them a product that's going to do the best for you in your crop. So once the nematode box arrives, you do need to deal with it immediately. So you do have to open it up. You want to take a look at those cooling packs. You want to see if there's any coolness that's left in them. You also want to take a peek at the shipping container that it came in. You want to make sure that there's been no breaches in that shipping container, which could prevent the coolness from happening in the container, but also letting warm temperatures to come in. I always encourage people to take a look at using a dedicated refrigerator. We don't want to, the worst thing you could do is have a staff refrigerator for your nematode storage because that door is getting opened and closed how many times in a day. That allows big temperature swings to happen, which means it could affect the nematodes that are being held in there. And then at the time that they arrive, confirming viability. It's the best to do it when they arrive. Lots of growers do it at the time that they mix, but I like to do it at the very beginning as soon as those nematodes arrive at your facility. A simple viability check, and there is, by all means, there are a lot different ones or a lot more technical ones. This is the one that I use on a regular basis. I take a small amount of the nematode product. It may come in a carrier material. It may come on a sponge. I mix it with tepid water. I put it in a shallow dish. I allow it to sit for 5 to 10 minutes just to allow those nematodes to rehydrate and start to get moving in there. And then I take something as simple as a hand lens, something just like a little guy like this. That's all that you need is just a small hand lens. I put it on a dark background, and then I'm able to actually take a look and see the nematodes in that liquid. You can use a microscope if it's available to you. I ask that you just use some bottom lighting. It just makes it a little bit easier to see them. Live nematodes are S-shaped or C-shaped or J-shaped, so they clearly have a shape to them, and typically they are actively moving through that liquid. Dead nematodes are straight, they're rigid, they're motionless, very different from what a live nematode looks like. As an example, on the left-hand side, we've got a dead nematode, so you can see it's straight, it's rigid, it has that very distinct straight shape to itself. On the other side of the slide, we've got some beautifully S-shaped, C-shaped nematodes. They look like they're actively swimming in that liquid. Those are our beautiful alive nematodes. Storage conditions. This is really important for protecting the viability. Nematodes need to be stored cool. The typical temperatures are between 4 degrees Celsius and 10 degrees Celsius. It can be very species dependent, so it's important that if you have a certain species, you need to know what the ideal storage temperature. I just gave you a range of what you can use. Do not freeze your nematodes. It is devastating to them. They do not do well with any kind of freezing conditions. And as I said, you really want to think about having a dedicated refrigerator whenever possible. Again, we're really just trying to prevent those huge temperature swings when you're storing these products. Most important thing is to look at the expiry date when the products come in. Make it a part of your procedures that the first in products are the first out products in your inventory management system. We don't want nematodes that are sitting around that become expired before you have the opportunity to use them. By no means at the expiry date do all of those nematodes die in that package, but they start to lose their viability. It is incredibly important to look at expiry dates and to use them before that date has come. A couple of do's and don'ts when you're getting ready for an application, just to keep in your mind when you're thinking about using this product, is you want to have areas where you can take the nematodes out of the fridge, allow them to warm up to room temperature before you mix them with water. Really important that we don't shock the nematodes from taking them from a very cooled environment and putting them in something that is like a tepid water. It can be very shocking for them, so it's important that you give them time to acclimate before you start to mix them. When you start to mix them, the clock is ticking. If you mix them with water for your application, you have about 4 hours to get them down. Ideally, I like to tell my growers, try to get it down in 2 hours. So as soon as you start to mix them with water, that's the time that they need to start, the application needs to start. They need to go into a tank that's agitated. This keeps them within the water column. They're heavier than water. They can sink to the bottom. So it's incredibly important that you have a tank that gives you agitation to keep them in the water column. You also want to use clean water with them. And then you want to look about compatibility and the inputs that may affect the nematodes. So we want to make sure that we have some idea of products that are compatible with nematodes and ones that are not compatible with nematodes. Some of my don'ts are, again, we want to don't want to mix them with incompatible pesticides. Think of those harsher chemistries. Organophosphates is a good example. That is not a great mix for our entomopathic nematodes. There are a lot of compatible products that you can mix with nematodes, but you do need to be able to take a look at what you're using before you start this application. You also don't want to leave them sitting in that tank for an extended period of time. Like I said, the clock is ticking. You have about 4 hours to really get them onto your crop and out of that tank. Combining them with harsh agrochemicals without checking is an absolute no. Compatibility is going to be an issue with these ones. And then nematodes are sensitive to the environment. We don't want to let them dry out on the surface of the soil or on the foliage of a plant. We don't want them to overheat, and we don't want them to sit in direct sunlight. All of those are devastating to nematodes. When and where to apply them. Ideally, you want to match your nematode to the most affected stage of your pest. So think of nematodes as going after the young stages. So your larva stage, your nymph stage, even your prepupal stage when it comes to thrips. You want to match that stage to the time that you're doing your nematode application. Placement is incredibly important. We want to make sure that we are applying the nematodes in the area where that pest is feeding, that pest is residing. It's really important that you make sure that nematodes go where the pest is. We want to look at our crop stage. Nematodes take time to affect the pests that are in your crop. If it's a crop that's going to be pulled out in a week or so, this may not be the control for you. We also want to look that we can get to the root zone with our application. Is that root zone applicable to get water in that area? Really important to take a look at that. And then also even the microclimate of that root zone, making sure that the temperature isn't too high. Nematodes can handle temperature ranges between about 15 to 25 degrees Celsius. What's the microclimate where I'm actually applying? Does that microclimate have enough humidity? Because nematodes are very affected by humidity. And then we want to make sure that we can give adequate moisture. Like we've said, they need that moisture to move through the soil to get to where your pests are. We have to have a soil that is moist we like to think like a little well wrung out sponge is a good sort of analogy, but we want to make sure that that soil has some moisture and that the soil temperature can support our nematode movement. Again, they have a range between 15 to 25 degrees Celsius for a soil temperature. Choosing an application method. Nematodes can go through all kinds of equipment. They are pretty versatile in that sense, but you can do applications in two different ways. You can do a broadcast application to give coverage a little bit everywhere. Think of doing across a production range or across a turf area. You can also do targeted applications, so just a hotspot of pests, or in specific containers, a crop that is just really needs that nematode application just in that certain crop. In our greenhouse, it's usually done as a drench. You can use an injector, a hose and sprayer. Even a watering can work as an application method for you. I caution with all of those, we want to make sure that they are going down with enough water to get into the pots to the point where we have a good drench, but not to where you're getting runoff. If you're doing a field application, you can use conventional equipment for your soil application. You can do a sprench, which is a drench and a spray mixed together. It can also go through an irrigation delivery system. Nematodes work with all of those. Some practical tips on your application, and these are really, really important to use when you're doing this, is you want to remove your fine filters and your screens. Nothing smaller than a 50 mesh or finer. This actually can clog as the nematodes are going through it, trapping them. So again, they're not going out onto your crop. It can also, due to the pressure in these equipment, can shear them as they're trying to get pushed through the screen. The best advice is to, if you can, to remove them from the equipment so that you're not having this sort of problem. Also, you want to have a large nozzle opening. 0.8 millimeters or bigger. Think of lovely water droplets, not a fine mist when you're applying them. Keep the pressure low. Now, I say 72 psi. Low is better than high. Nematodes can withstand much higher pressure than that, but keeping it low is definitely going to guarantee that they aren't going to be affected by that. And then avoiding any kind of piston pumps, only because they can build up the pressure over time, which then down the road as you're applying could give you some problems with it. You do want to maintain continuous agitation. They need that agitation to keep them within the water column so that they are coming out at an even rate onto your crop or your area. If you don't have proper agitation, they sink to the bottom of the tank, and basically the first part of your application, you're getting no nematodes out, and at the end of your application, you're getting all of your nematodes out, which doesn't give you a very even crop distribution, and then obviously not even control. The environment, the rate, and the monitoring. Most important thing when you're thinking about a nematode application is you want cool and low light conditions. Think of cool temperatures, think of overcast skies, late in the day applications. You don't want to have any sort of bright conditions, high UV light. All of those are devastating for nematodes. We want to keep our soil and our media moist before the application. Again, they're using that little water channel to move down to where the pest is. And then we want to keep that area moist after the application as they hunt for those pests in the soil. They need the water channels to be able to do that. You need to follow the label rate. Every company that produces beneficial nematodes has their own unique label rate. So I won't speak very much about that, but you do need to follow the label rate. The worst experience that we have is growers that have a really high pest pressure opt to double the label rate or triple the label rate, thinking that they're going to get much faster control. What they find out very quickly is that they get almost no control. Nematodes can communicate with each other. They can send out signals, usually through pheromones, that there is too many in an area and they infest nothing. So you have to follow the label rate. Don't have the idea that I'll just double it to get faster results. It doesn't actually work that way. You may need to adjust and think about not doing the doubling, but doing a second application instead. That's a better way of using them. Always really important when you do an application to check your pest activity after the treatment, just to see how well things have gone. And that gives you a way to be able to decide if I need to do that second application. My idea with nematode applications is it's the best nematode application is the one that places live nematodes. So we've stored them correctly. They're live in the right place. We know where our pest is in this crop and applies them at the right time. It's a beautiful overcast day under the right conditions. It is not too hot out there. So keep that in mind when you're thinking of these applications. Just a few reminders of the common mistakes that you may come across. Storing your nematodes too warm. So remember, it is 4 to 10 degrees Celsius. For too long, we didn't check that expiry date, and freezing them. Applying to media that is dry, hot, or in bright conditions. Nematodes very sensitive to high temperatures and UV light. Using spraying equipment that has a fine filter, a tiny nozzle opening, and also giving excessive pressure. All of that will cause problems for you. Mixing your nematodes and letting them sit in that spray tank before you apply them. Nematode applications is not, I mix it before lunch, go to lunch, and then come back in the afternoon and apply them. No, once you start mixing them, that's the time that you need to apply them. Mixing them with untested pesticides or incompatible biologicals. It's incredibly important to look at that compatibility before you do your applications because it can cause you a lot of problems and it could make it that the nematodes are not working as well as they should. Also applying the pest at the pest stage where it's in the root zone. So making sure that that pest is in that root zone where you're applying these guys. What drives success in any kind of nematode application? Scouting early and monitoring consistently. With any IPM program, those are the two things that you really need to do. You need to make sure you know what's going on with the crop and the pest that you're dealing with. You want to make sure that the pest is vulnerable. So it's in that larva stage in the soil. You want to match your species of nematodes with the crop, the pest, and the habitat. Every nematode species is unique to those types of conditions. So you want to make sure you're using the species that works best on what you're dealing with. Combining nematodes, though they can in some cases be a standalone, but also combining them in your program with other cultural practices and using other biological controls at the same time that you're using the nematodes to give a full control program. And then looking at compatibility with the chemistries. We don't want to use products that are going to be damaging to the nematodes when we're putting them out into this crop. A couple of metrics to measure as you're doing nematode applications and really trying to understand how well that they're working. We want to do our pest counts before and after a treatment. I love a good record keeping moment. So lots of notes on how many pests we have, how many pests that are in the soil, how many pests are on sticky cards. All of that is incredibly important. Then we do want to look at plant damage over time. Is the new growth coming out cleaner than the previous growth? Could give you a good indication that the nematodes are absolutely giving the controls that you need. We want to know how moist that soil is and what the temperature is in the soil and outside temperature during that application. That can give you some ideas of, hey, maybe I might be able to improve this if this couple of degrees of temperature outside is a little bit cooler. And then understanding that we may need to do repeated applications at regular intervals to gain the results that you're looking for. Just a little practical example. A grower finds this. So they find this yellow sticky card in their crop during their regular scouting. It's loaded with what is fungus gnats. Here's a simple decision, a tree for the grower to use. So he scouts his crop. He confirms that we have the pest on the yellow sticky cards. He goes to the pots of these plants and starts to look for the larva that's present. He identifies that pest as fungus gnats. He then looks for the correct nematode species for fungus gnats, which would be something that has Steinbernema felchae in it. The grower gets these in stock. He looks for the best day to apply them. So he wants to make sure that the soil is moist before we do it. It is a day that is overcast. There is no direct sunlight at all. They mix it up. They apply it as a soil drench or a spray. They water after the application to allow the nematodes to move through the soil. And then they monitor for the next 2 weeks just to make sure that things are going well and that the nematodes have that water in there to be able to get to this pest. And then they redetermine at that time, hey, do I need to do a second application, or has this application worked well on the pest that I was targeting it for? Successful nematode programs are built on scouting, timing, compatibility, and follow-up. It's not just luck in any case. It's really important when you're thinking about using these that you have to have all of these elements into the mix to have a successful nematode application. There's going to be failures. We can't get around it. Some of the failures that we see are improper storage. So we had a fridge that failed and the temperature got to 15 degrees Celsius for a week and we didn't notice. That's going to cause a problem. There was a delay in application and mixing or delivery. All of those, we let them just basically sit in the tank for too long before we applied them. We use an incompatible pesticide, or we decided to take a shortcut and tanks mix everything into that tank with the nematodes, thinking that it will make it easier, and there's some incompatible products in there. There was a poor timing. We chose a day that was super bright, or it was incredibly warm, so the environment wasn't really great for them. The pest was simply at the wrong stage. It wasn't a larva in the soil. It was an adult that was out on the foliage, or we opted not to apply them to the right root zone, exactly where the pest was found. When diagnosing the problem, I start with some simple questions to my growers. I start with the basic one, which is, how did you store them? If that was stored correctly, then we start to look at, was it applied at the right time and in the right place? So was that pest present in the soil, and did we apply the nematodes to where the pest was present in the soil? If that's also a yes, then I move on to the equipment. Was there anything in the tank? Was there anything in the water or the environment that may have reduced their survivability? All of these questions can kind of help you to understand what went wrong with a nematode application when it doesn't go the way we think it will. We can turn these failures into a learning experience by keeping detailed records, understanding the nematode species that we applied, where we applied it, when we applied it, what was happening in the conditions that were out there. All of that can give you clues on what to do the next time you do an application to improve it. Comparing your treated or untreated areas may be giving you clues, again, to the environment or where the pest is at that time. Maybe that's the reason it didn't work as well. We can track our temperature, moisture, and pest pressure. All of that's incredibly important. And then all of this bits of information can give you an idea of how to improve your next treatment with them by sort of keeping that record keeping and understanding what may have gone wrong. What I really hope from this small part of the presentation that you take away from you is understanding how to protect these nematodes from stress before application and during application. Keep in mind the product that you hold in your hand could have taken a company weeks to manufacture. I know for us, there has been years that we have developed nematode species to provide for our clients. So it may have taken a researcher years to be able to provide the product that you hold in your hand. So please think about that when you're storing and handling them. Did I apply under conditions that support their survival and movement? Did I take a look at the environment that I was using them in? Did I match my nematode application to the pest life stage that I'm trying to manage? That's really important. And then the success of this is going to basically give you the results that you need, and it's not going to be guesswork. It's just going to be part of your day-to-day routine when you're using entomopathic nematodes. Thank you all so much for this opportunity to be able to speak to you about this. It was an incredible honor to be able to do this, and I'm going to send this over to our next presenter, which is Dr. Diana. Thank you so much.
Thank you so much, Stacey. So I'm gonna share my screen. So shared. Did you see it?
Yes, we can see it.
Okay. So thank you so much for this invitation. I am really pleased to present today about beneficial nematodes. My name is Diana Londoño. I work in BASF. I am the global technical lead for bioinsecticides, that includes the beneficial nematodes. I including here my email in case that you had any questions. Please feel free to contact me. So first of all, I'm gonna talk about what is the purpose to use nematodes in the integrated pest management programs. As you know, when you talk about integrated pest management, that includes different combinations that can be cultural, that can be mechanical, behavior, chemical composition, and also biological, time of the year, time of the insect. So there are a lot of things that come together. When you're gonna think about IPM programs, you have to think about what are the most important benefits or qualities that have the beneficial nematodes. Here in the right side, I have different kind of nematodes. For example, I have Feltiae, Ceronoia feltiae, that is very good nematode in temperatures between 15 Celsius to 22 Celsius. But when I talk about crusade, this crusade nematode start being active since 5 Celsius. So you have to start keep in mind what insect can be good for this kind of nematodes depending on the temperature, for example. Or we had Stenonema carpocapsi. This nematode is working better in dry conditions and also is working better at very high temperatures, higher than the Feltiae, higher than the Crusade. We are talking about 25, 28, even 30 Celsius. Also, we are talking about the Nemaslug. This is a different kind of nematode. There is the Phasma californica. This is a specific nematode to control slugs. So the behavior is a kind of different. Also, the size of the nematode. And I'm going to talk a little bit about the IPM program that we use for the nematode. So important things that you have to keep in mind when you're going to choose a nematode. They are working very well in the right conditions. So you have to think about the humidity. I'm going to show also some examples about the temperature, also discussing about the temperature that is good for this kind of different nematodes. And also keep in mind that these nematodes that have a different behavior. Some are crusades, so they like to follow the prey other than ambush. So that also is important when you're gonna choose the kind of nematode that you're gonna use. The other thing that you have to think about it is the right host and the instar. I'm gonna give also an example where we choose the right instar to apply the nematodes. It's persistent in soil if the insect is present. And also one important thing when you're gonna use IPN is safe for crops. Yes, it's not producing phytotoxicity, is very good for the environment and also for the users. It doesn't require the re-entry interval and doesn't leave any residues. Important thing is application methods. You don't need a new equipment, anything that is fancy. It is easy to apply by irrigation. It's easy to apply for a sprayer. And about the compatibility, this is a very good, important point. Yes, every company has a list of compatibility. So in our case, we have the compatibility with different kind of chemical products, including fungicides, herbicides, insecticides, chemicals, insecticides, and also biological ones, also adjuvants. This is a very important key that I'm gonna touch at the end, because in that way you're gonna be able to know if you can use tal mix, if you have to apply the next day, if you have to wait some hours, maybe apply in the following week, or definitely you are not able to apply together at any time. For the beneficial nematodes, one thing that I find that is very good is that can be used in the conventional, because sometimes it's the only product that is working in a specific instar, and also can be applied in the organic farming. I'm going to start with my first example of the IPM program. So I'm going to talk about the golden moth, Cydia pomonella, that is an important pest that is attacking the pears and also the apples. We had different programs, one that is in the US, in the Pacific Northwest, other one that is in Europe. Europe include different countries, like for example, the UK. So you have to think first, what is your region? For example, in the UK, we have around one cycle per year for the insect. While in the US, in the Pacific Northwest, you can have between 3 to 5 cycles per year of the insect. So that means that the application timing is going to be different. Also, the amount numbers of nematodes that you're going to use. So this is very important to keep in mind because the number of generations is very important when you're going to think about the IPM program that you're going to use. So here, this insect is very interesting because you have, of course, the adults, they're going to lay the eggs and they're going to start having the first instar. So you don't want to apply the nematodes at the first instar, where are the leaves. The nematodes, they are not going to survive the dry conditions in the leaves. So you need to apply the nematodes is in the trunk or in the soil. So the first instar of the insect is not a target for the nematodes. So when they set the first instar, they go and attack the fruit. Definitely, you're not going to apply the nematodes with this instar because they are already, the insect is inside the fruit. So the nematodes, first, they are not going to survive in the dry conditions of the fruit. They are not going to penetrate the fruit to attack the insect. And when the insect is inside the fruit, definitely the fruit is already wasted. But what happened after? You had the overwinter larvae, that is that we call. This overwinter larvae, is in the soil and also is in the trunk. Here is when you apply the nematodes. The nematodes are very efficacious against these larvae, and also they are efficacious against the pupae. So the next generation, we're gonna see the decrease in the number of nematodes. Here, maybe it's gonna be easy to see. You can see when they had the different larvae and when you apply here the nematodes. So you can see here between August to December, you can apply the nematodes. Here in the US, we apply from August to October, for example, when you can apply when the overwinter larvae is there. And the temperature of the soil is very good for the nematodes. Here, I want to show you the life cycle of the nematodes in the cold moth larvae. These nematodes are very efficacious against these larvae, as I told you. So you can see here when they find the insect and they can enter by any hole in the insect. Here is a picture of the nematode entering to the spiracle. After that, you can see that the nematode enter and start releasing the bacteria. This is the mode of action also that they had the nematode. So the nematodes eat the insect and also release the bacteria. The bacteria help to kill the nematode and also give a food for the insect. I'm sorry, for the nematode. So in that way, they can continue multiplying. You can see here a bigger instar of the nematode that is already adult and start the second and third generation. And after that, destroy the larvae. You can see here, everything destroyed. So the nematodes go out and start looking for new hosts. We did different studies. I'm going to show only the study that we did, one of the studies that we did in the Pacific Northwest in Quincy. Why we did the application in spring, summer, and fall. Why we did this? While I was talking before that we had to do the application only in the fall, is because, as I mentioned, you can find over there between 3 to 5 generations of this insect. So what is happening? We found that it's very good when you do the application in the spring and in the fall. Summer, also you can have a good mortality of the insect, but however, keep in mind that summer is very hot, is very dry, so that decreases the chance of the nematodes. Also, one important thing that you have to keep in mind is the number of nematodes that you do for application. So we found that 750 million per acre was very good for that region. Now let's move to a different nematode. So here the other nematode that we have is the Phasma californica. This is a totally different nematode. It's for the slug control. We had this product only in Europe. And one thing that you have to keep in mind is that the host, yes, it is not working against all kind of slugs. That is very important because sometimes you have in mind, oh, that is working with the slugs, I'm gonna apply, but it's not the target that we have for that nematode. So we had different kind of studies. We are recommending this especially for the Drosera reticulatum. And this is also very important to keep in mind. I'm going to show here in this slide the life cycle of the slugs, and it's very important to use the nematode when the slugs are small. So we are talking about juveniles. So they are small. You're going to have the application between March and October. During the year, you can do application weekly. So you use 50,000 nematodes per square meter, but if you use every 2 weeks, you have to use 150,000 nematodes per square meter. But if you decide to do it every 4 to 6 weeks, so you have to use 300,000. So the number of nematodes varies with the number of applications that you're gonna decide. And yes, this is the Phasma californica. You can see they're moving. I want to show one of the important studies because we were talking about the temperature. Yes, when you're gonna do a integrated pest management program, you have to think about what is the right temperature to use it. So here we find that the 16 Celsius, yes, or moderate temperatures are the very good for the nematodes, while higher temperatures are not good to control the slugs, for example, in this study. Another one is when we start looking for the humidity. Yes, when you find the humidity on the soil and the kind of soil. And you can find here that we have better results in the siding loamy or soil than in the coarse substrate. So this is also to keep in mind because when you're gonna apply the nematodes, sometimes you are thinking, okay, didn't work in the way that I wanted, but I apply in the right temperature, maybe no. I apply in the right humidity conditions, maybe no. So it's something that you always have to evaluate. So what are the conclusions, for example, for the slug control? One here, that the nematode can be used in the IPM program. Yes, they can be used in the wet conditions, something that is not working with the ferriphosphate pellets than they are the chemicals. You can use both and you're gonna have a better control for the slugs. And also you can use an nematode here as a preventative. So this is something that you can evaluate in your place and decide how you're gonna use it. The third example that I have is the Western flower thrips. This is a lot of questions all the times. I can use the nematodes against the thrips. The answer is yes. So we have here some pictures of the nematodes attacking the thrips. These pictures are generated in our lab in RTP, North Carolina, where you can find the nematodes entering to the thrips, attacking the thrips, and also multiplying in the thrips. So when you apply the thrips, you have to think that you're gonna target is the insect that they are in the soil. Yes, so we are talking about the immatures and also about the pupae. The adults are very difficult. So if you apply the nematodes in the foliar, you have to try to protect the nematodes. So sometimes we use it some adjuvants that give a kind of protection for some hours, but definitely the best application is in the soil. So here I want to show now is when we did comparison between chemical programs and also these nematodes programs. With the chemical programs, we did this trapping number of the adults, and we found that at different weeks, you have a lot of, you can see here, males or females, and they are trapping, while the next slide is showing when we apply, for example, the nematodes in the right conditions, and you can find here that definitely the decrease of the number of adults is notorious. So when you apply the nematodes and the right conditions, they are going to work very well. So one thing that I want to talk is about the compatibility. Compatibility is very important. As Stacey was mentioned, it is no way that I'm going to mix with this to see what is going to happen. You have to have the information. As I mentioned, different companies, as for example, BASF, you can find this information, yes, in the website. When you can start looking for your product, can be a fertilizer, can be an adjuvant, can be an insect, sometimes ask me about beneficial insects or fungicides, and you're gonna see, okay, it's good to apply, for example, here in Talmix. But if we go, for example, for this product that is and it's aligned, you have to apply separately because it's gonna basically no very good for the nematodes. Or we have different ones, for example, here, the Rubigan, apply separately, 7 days apart. So you can have all this information for the nematodes. One thing that is, to me, very important to keep in mind, doesn't mean that if for Stereonema feltiae they are compatible, you're going to assume that for Phasma californica they are going to be also compatible. Or for example, you have Stereonema feltiae is compatible and Stereonema cruzae can be because both are Stereonema, but sometimes the behavior of the nematode and also the temperature and also the formulation of the product can have some different answers. So please, my advice is always, always check the compatibility information. Yes. So compatibility things that I want to show here is, as I mentioned, with the formulation that change, with the dose that you're using, also is very important to keep in mind that you're using the right dose for your chemicals or biologicals. And one important thing also is that some information for some companies I found is for the IOBC in the rating. So you can find here if they are 3 or 4, basically they are not compatible, but if they are 1 or 2 or 0, they are compatible. And one final note is about the adjuvants, because many people are thinking, okay, if I use adjuvants, they're gonna work wonderful. Please look the adjuvant information. Because not all adjuvants are good for the nematodes. Sometimes they help the nematodes to dry quickly, as I mentioned here. Some are very good in the lab, but when you apply in the field or in the greenhouse, they are not so good. There are some adjuvants that can improve the control. As I was mentioned, they kind of protect the nematode and keep the nematodes alive for longer time when you apply in the foliar. And definitely, please always check if the nematodes are alive or are dead. We are using this device for the cell phone that is working very well because we know that not everybody has a microscopes in the field and also, of course, in the greenhouse. So this is very useful, and you can find a very good information if you have good number of nematodes moving very good and they are not dead. So with that, I'm gonna stop here. So again, thank you very much for the invitation. Yeah.
Thank you, Diana. I'll take back the floor. Thank you very much for all of our speakers for your presentations. We've had a very large number of questions, great engagement from everybody on the call. So thank you very much. And I'll just invite our speakers to take a look at those open questions that are still in the Q&A and answer them if you can. In the meantime, I'll talk us through some of the questions that came up in the chat. So one question was, are there any statins of EPNs which are resistant to commercial nematicides? Who would like to take that one?
I think I answered that.
Oh, brilliant.
But just to say, like, I have never come across some, and maybe the others have, that EPNs that are resistant to nematicides, but there was one study showing that it's feasible to genetically select EPNs that would be resistant to nematicides. Edamon Glazer's lab did that. So if there's enough commercial interest, maybe they would follow that and have a resistant nematode. Nowadays, I guess most people would just recommend don't apply your nematodes at the same time or close to the same time that nematicides are being applied.
Thanks, David. Another question that's come up in both the chat and the Q&A is on the possibility of using nematodes in aquatic habitats, particularly in the case of control of mosquitoes. Would anybody like to take that one? Maybe Stefan or Diana?
Yes. First, the nematodes are for the soil, yes, and they need this kind of humidity. So basically, if you're using this aquatic conditions, the nematodes are gonna go to the sink. Yes, so they are efficacious against the mosquitoes. Yes, we found efficacy of the nematodes against the mosquitoes, and but the thing is, when you use the nematode, they had to mix all the time, so in that way they are gonna be distributed in the good way. You don't want the nematodes to sink with the because they had to look for the larvae. So also we found out when you it is possible, for example, to use if you are thinking to use in the house in these pop areas when you have the water and maybe some mosquitoes growing. But when you're gonna use in the field conditions, so you have to think about other situations and also here enter the cost of the nematodes because the nematodes are not cheap. So is you have to have the balance. So we are not
Okay,
suggesting now the nematodes for the mosquitoes at this moment.
thanks, Diana. I'll take one more question from the Q&A because I'm conscious that we are coming to the end of our session. Um, so one question is maybe for Stacey in this case, what's the normal shelf life period of EPN products after harvesting or packaging within require the cold storage condition? If an expiry date of EPN products is coming up very soon, does that mean that the products were already old before shipping to us?
One question. The typical storage is for the nematodes that we produce is about 3 months in refrigeration. If your expiry date is coming up to the time, they potentially have been processed or reared a lot sooner than when you receive them. So that may be the case in that instance. But like I said, expiry date is a guideline. They don't all die after the expiry date, but it is really important to try to use them within those dates.
Thanks, Stacey. Okay, there are quite a few more questions coming in. Again, thank you so much to the audience for your engagement and for being so proactive and asking these questions. We will be sharing the recording of the webinar, and through that, maybe some recordings of the Q&A as well. And so you'll be able to see some answers through that, we hope. But for now, I'm afraid we do have to close the session. So thank you again for taking the time to join us, both the audience and our speakers. We've had a really excellent overview of how beneficial nematodes are used as effective biocontrol agents and coming from all angles, from research all the way through to the field. So thank you to Dr. Stefan Toepfer for setting out the scientific foundations and what that means for their use as an agent, and also for talking us through how you can use the CABI Bioprotection Portal to find registered EPN products. Thank you to Dr. David Shapiro-Ilan, who talked us through some case studies and also demonstrated some improvements that can be made to continue to make EPN products more efficient and improve their efficacy. Stacey Hickman from NIC, thanks for all of your practical advice on handling storage and application, which I think will be of great help to everybody who's using EPNs in the field. And thanks also to Diana Londoño from BASF for describing how specific products work and, importantly, how they can be used within an integrated pest management approach as well. So we hope that you found this session as interesting as we have. As I said, we will circulate a recording to the registered attendees so you can keep the information close at hand. Don't hesitate to reach out to us if you have any further questions. And until then, thank you again and have a lovely rest of your day. Thank you and goodbye.
Thank you.